Is everyone ready? Shall we start? Yes. Okay, listen. So today we will discuss about the estimation of hemoglobin. So far how many practices we completed? We completed ABO grouping. I hope you know everything about ABO. And we have completed ESR. Any more doubts in these two practicals so far? No, right? What are the methods of ESR? Western rinse and retrodes. Which one is lengthier? Longest use? Western rinse. And who discovered ABO? Carl Landstein. Universal donor?
Yes, sir.
Universal donor. O negative. Okay. Universal recipient? AB positive. Very good. Okay. Very good. O negative is correct. I was just thinking. What is the color of RHD? Colorless. Okay. RH factor discovered in recess monkey. Very good. Who discovered RH factor? Alexander Meier. Okay. Purpose of ESR? Any two diseases? Any two inflammatory conditions? Rheumatoid arthritis and yes, MT. Very good. So next one is HB estimation. What is the purpose of HB, by the way? Hemoglobin purpose. To carry oxygen and carbon dioxide, right? So where does hemoglobin located? Location of hemoglobin? Surface of RBC. Very good. RBC surface. Very good. Then what is the normal value of hemoglobin? Normal value of hemoglobin per cubic millimeter of blood or deciliter of blood. What is the normal value? So write down males and females. Normal HB in males is around 15 to 17 grams per deciliter. Okay. Keep in mind this is in grams, not milligrams or micrograms. HB is always in grams. Keep in mind. Okay. Grams per deciliter, 15. Or in some textbooks they will say 40. That is also normal. Plus or minus 2. Plus or minus 2 means more than greater than 2 or less than 2. And for females this is around 12 to 15 grams per deciliter. Values are not absolute, they are ranges, and textbook to textbook there will be slight difference between these ranges. But this is the normal range of HB. Clear? Yes. So why blood is in red color? Because of presence of hemoglobin. So hemoglobin associated with iron binds with oxygen, rusting process leads to red color. So red color is due to hemoglobin in a nutshell, right? Yeah. So now we are trying to estimate the levels of hemoglobin. What is the purpose? Why we need to measure levels of hemoglobin instead of counting of RBC? Just we can count RBC and count of RBC will be directly proportional to HB levels, right? Am I correct? Because HB always present in RBC, then why we are separately measuring HB rather than relying on RBC count? Why we are yes, Melissa.
Yes, sir.
Yes, miss.
Because patients can have hypochromatic anemia. So
Very good. Well done, Melissa. Listen, since you have red blood cells, doesn't mean they have hemoglobin. Sometimes HB content might be reduced on each red blood cell. This is red blood cell. Okay. So that is why not only red blood cell count, but also we need to count the HB content per deciliter of blood. Sometimes what happens? HB will be less than the normal value. Less than normal value, and sometimes HB will be more than normal value. Okay. Less than normal value is called as, less means hypo. What is the medical term or scientific term for color? Chrome. So hypochrome. Okay, it means to be anemia. Anemia. Decreased HB count leads to a anemia called as hypochromic anemia. Decreased HB count leads to hypochromic anemia. That means less colored anemia, right? Then opposite, more HB value greater than normal value is called as hyper. That's it. Hyperchromic anemia. Okay. Now let us see in what conditions hypochromic anemia might happen. Conditions where hypochromic anemia might present. Yes. Yes. Any idea? Yes, Melissa.
Iron deficiency.
Very good. Excellent. Everyone appreciate her. Chapter don't sleep in the class. Please don't sleep. Yeah. So very good. What is that? Iron deficiency. Iron deficiency lead to hypochromic anemia. So that is one of the dietary factor. So if your diet don't have appropriate iron, then that leads to hypochromic anemia. Okay. Any other factors leads to hypochromic? One was iron deficiency. Any others? What if RBCs are lysed? Somehow red blood cells are destroyed in the blood. That lead to hypochromic or not? So destroyed RBCs will be destroyed. The corresponding HB also will be degenerated into bilirubin. So RBC lysed conditions can also lead to hypochromic anemia. Now can you give me some causes of RBC lysis? What causes lysis of RBC? Any one condition? Causes of red blood cell destruction. Multiple causes are there, by the way. Yes, Miss Melissa. Do you want any clue? Yes.
Toxins in the body poison.
Toxins are very general, broad thing, more specific. Of course, some toxins will lyse the RBC, I agree. Any more? Okay, very good, Ria. Maybe mismatched blood transfusion. Mismatched blood transfusion, immediately there will be lysis of RBC, decreased hemoglobin, that is one factor. Any other factors? Can you give? Can anyone give one infection mediated RBC lysis? Microbe mediated RBC lysis. There is one celebrity parasite that is extensively feeding on RBC. You already know the answer, by the way. What parasite? I think you did prove again. Mosquito. Now tell me the answer. Malaria. Very good. See, everyone thinks that malaria caused by mosquito, but it is caused by a parasite called falciparum. Right? Plasmodium falciparum, vivax, oval, and malaria. So malaria parasite, malaria infection, hypochromic anemia. Any more conditions? HDN. Have you heard about HDN? Hemolytic disease of newborn. Okay. Just born babies will have jaundice. You might heard it, right? Just born baby has jaundice. That is called HDN, where the mother antibodies attack the baby red blood cells. That leads to this condition. In that condition also, this can be seen. And of course, pregnancy. Okay, in pregnancy, definitely women tend to have anemia due to extensive feeding by the fetus. Actually, blood was deviated to fetus, so she will be slightly anemic. That's why we are giving her diet of folic acid. Folic acid is one of the important factor responsible for hemopoiesis, if you remember, or erythropoiesis. Can anyone say what is erythropoiesis? Production of red blood cells in the body. You remember? Yes. There are multiple erythropoietin, folic that's it. So anyway, so now you understood the significance of hemoglobin, right? Hemoglobin directly or indirectly connected to anemic condition. Not only your red blood cells, but hemoglobin also plays a vital role to check the anemic status of the body. Is it clear? Now how to detect this hemoglobin? Methods of detection of hemoglobin. Hemoglobin detection methods. So to understand the methods, first you need to understand various states of hemoglobin in the body. Sometimes hemoglobin is associated with oxygen, that is called as oxygenated hemoglobin. Then hemoglobin associated with carbon dioxide, this is called as carboxylated hemoglobin. This is, to be precise, this hemoglobin is called as weak hemoglobin. Weak hemoglobin. There is a methyl group that will attach to the carbon dioxide with hemoglobin. That is why this is called as weak hemoglobin. Okay. Then so far I said HbO2, HbCO2. Then there is another method. This is weak hemoglobin by the way. And the third method, colorimetric method. Colorimetric method. So here we are using Bayes and Lambert's law. I hope you already know Bayes and Lambert's law, right? Bayes and Lambert's law. Do you know what is it? You don't know. Do you know what is colorimetric method? You don't know. You haven't studied it, right? Okay, there is a small instrument that will measure the color of any substance, color of reagents. Based on the intensity of color, it will give the reading. Okay, that is called colorimetric method. And fourth one, automatic method. Automatic method. And last and final but most important method, that is our Sahli's method. Sahli's method. Sahli's method is also called as visual method. Visual method. HB estimation method. Visual HB estimation method. So now you understood the methods of HB estimation. Colorimetric method, automatic method, and Sahli's method. Is it clear? Yeah. So I just want to add a few more things in colorimetric since you said you don't know. Very simple idea. Imagine I have a water and I have a turmeric powder. Okay? I added 1 gram of turmeric powder to the water. Now what will be the color of water? Pale yellow. Now I added 10 grams of turmeric powder. What will be the color? Strong, okay, deep yellow, okay, intense yellow color. That means the color of reagent is directly proportional to the number of solutes present in the water. In this case, turmeric powder. So if you measure the intensity of the color, intensity of the colored reagent is directly proportional to the concentration of the substance present in the colored substance, colored water. Am I correct?
Yes, sir.
Now I just say concentration is directly proportional to intensity of the color of solution. The higher the intensity, the greater the concentration of the substance present in the colored solution. Is it clear? This is the principle behind colorimetric method. Okay. Now, this is one of the standard and accurate method, but still, colorimetric method will do some variations because sometimes hemoglobin will be associated with oxygen. Now, this will have deep red color and this will have somewhat bluish color. Due to this bluish color interference, we will get a deviated results. So the gold standard method is always cyanomic hemoglobin method. This is the gold standard method for estimation of hemoglobin actually. Okay. Then we have automatic method, very easy. Automatic method is just a hybrid of colorimetric method. By the way, this machine can also measure hemoglobin apart from RBC count, blood cells. Okay. It's a advanced machine anyway. So now, last and final, Sahli's. how Sahli's method is different from them. I'm saying again, Sahli's is not a gold standard method, but this is one of the most widely used, commonly used method. Can you guess why it is commonly used? Because this is cheap, easy to perform, and need less manpower to do this test. It is cheap and economic. That is always our first priority in healthcare. Affordability. First we should go with the cheap tests or simple tests. If these tests give any abnormal results, we will go for high-end tests. So first we will go with the simple test. Okay, simple and cheap test. I cannot say cheap, affordable test. So cheap will, it will degrade the significance. So affordable test. Is it clear? So now let us see how we will perform Sahli's test. So this was adopted by a scientist in 1870s. It's one of the oldest tests. So by a scientist called Sahli's, hence Sahli's method, right? So he came up with a very interesting approach. He said like this. So first he took blood and he want to convert blood to a reaction. He want to convert a standard reaction. He want to apply some standard reaction to a blood. For the standard reaction, the reason why I said standard reaction, because when we collect any 5 of your bloods in a test tube, all your bloods will have different colors, variations of red color, deep red, pale red, pinkish red. So if we have various red colors, then we cannot standardize this test, right? To get a normal standard color, we are subjecting blood to a reaction. In this case, the reaction will be, we are subjecting it to an acid. That acid is called as, we have standardized the acid. This acid is called as N/10 HCl. N stands for normal. Normality, molarity. Okay. So this is one normal parts of blood to acid. 10 parts of blood with 1 part of acid with 10 parts of blood. Okay. N/10 HCl. HCl stands for hydrochloric acid. So I have added acid to the blood. I know that blood has hemoglobin. Okay. I added acid to the blood. Now it will make this acid combined with hemoglobin, forming a substance called acid hematine. Within 2 minutes, you will get an acid hematine formation in the test tube. Is it clear? This acid hematine has a color. The color was brown color. It will produce a brown color, that is called as acid hematine. Okay, a deep brown color. We can say deep brown color. Okay. Now we are going to measure this brown color. We are going to measure this brown color. We are going to measure this brown color. Okay. So this is the idea behind the principle of Sahli's method. We are subjecting hemoglobin to an acid called N/10 HCl, leading to a product called acid hematine that is in brown color. Is principle clear to everyone? Yes. Now I will take to the next level. How he has standardized this test. Okay. So what he did was, he took a test tube, his own developed test tube. This test tube has a bottom marking that is 2. This test tube has a bottom marking that is 2. And this test tube has a upper marking that is around 22. None of the human will have 22 HD. Keep in mind. Okay? Grams per deciliter. Now pay attention. This is called as HB measuring tube. HB measuring tube. Now, in this tube, he will add N by 10 HCl. He will add N by 10 HCl. Up to what? He will add N by 10 HCl up to 2 mark. Up to 2 mark, he will add N by 10 HCl. Clear? We have added N by 10 HCl up to 2 mark. Next, to this we will add a specific amount of blood. This is first step. Then second step, we are adding blood to the tube. We are adding blood. So we are not going to add random drops of blood. Rather than we are adding a specific quantity of blood, that is 20 microliters. We are adding 20 microliters of blood to the acid. For this purpose, he has developed a standard pipette. This pipette is called as HB pipette. This is called as HB pipette. HB pipette. Now, to this HB pipette, there is a upper marking. That marking is 20 microliters. So, this is HB pipette. It has a upper marking that is 20 microliters. So, what you will do? You will pipette your blood up to 20 mark. You pipette it up to 20 mark and you will transfer this blood into the test tube. Okay. Then what you will do? You will wait for how many minutes? 2 minutes. Why are you waiting for 2 minutes? Now the blood will mix with the acid, leading to formation of acid hematite, that is brown color. Now, after 2 minutes, this is third step, waiting for 2 minutes. After waiting, what happened? Brown color has been developed. Brown color has been developed. Is it clear? Yeah. 1 second, let me keep it here. Yeah. Brown color has been developed. Okay. Next comes very interesting feature. This is a deep brown color. This is a deep brown color. Now, our Sahlis can take one coffee color filter. I'm saying coffee color filter. He has universally standardized this color. He has universally standardized one standard color. Standard color filter. Standard color filter. I used coffee, but the coffee is not the right term. You just imagine this is a comparator. This is called as comparator. Comparator. Comparison, for comparing. So what he did was, he kept 2 comparators in a stand like this. This is called a Sahli's stand. In this stand, can you able to see 2 comparators, 2 brown color rods? So he already kept brown color rods, and I will tell what he did with this. Now listen. We need to keep the test tube in this stand. Okay. Now we need to start adding distilled water to it. Drop by drop. Drop by drop, we will add distilled water. We are adding distilled water to this tube. Yes. Now, whenever we are adding distilled water, what happens? Deep brown color will gradually changes to less. I'm writing intensities here. Maybe I will write this as a 5-point brown color. 5-point brown color. So 1 point, 2 point, 3 point, 4 point, 5 point. At one particular point, at one particular time, when you are adding drops of water, at one particular time, the color of tube will matches with the comparator color. Okay? That's why I said 5 point. The color of tube will reach to the 5 point, that is equivalent to the comparator color. Got it? Until you are reaching same color, you need to add the water. You will add a drop of water, and then there is something called stirrer. I think stirrer we already broken it. There is a small glass one. So we added drops of water, then mix it. That stirrer we don't have, but you need to mix the tube, okay, in between, because it should be evenly mixed. So you have mixed it, then you will check the comparator color. Still if it is too much dark, you will add water again. So you will add water until where this color matches this color. The color of test tube will matches to the color of comparator. At one particular point, it will match. Once the color match, you take the reading. If this reading was 12, that will be your HB content. Simple. See how smart he was. So that's it. This is how we can perform HB estimation. So every one of us, we will achieve that comparator color at various levels of this height of this tube. And that high, upper meniscus value will be your HB value, because he already given graduations of HB content on the side of tube. So this is how we will perform the HB estimation by Sahli's hemoglobin method. Is it clear? So what are the components of apparatus? We need a HB tube, we need a HB pipette, we need a HB stand, then we need a stirrer to mix it, and we need a distilled water, and we need a reagent. What reagent it is? N/10 HCl. Until your death, you must remember these values. N/10 HCl, okay? If anyone asks what reagent we are using in HB, what is the reagent? N/10 HCl, right? What anticoagulant you used in ESR? Citrates. Okay? Don't confuse with these things. Color of EDTA? Violet. Purple. Very good. Fluoride? Gray. Fluoride is gray, okay? Citrate is black and oxalates also black. I think I have some doubt with this company. They gave black color for citrates. Usually citrate is for anyway. Any doubts in HB estimation? Who discovered this method? Sahli's method. What is the normal HB value in males? 17 grams per deciliter. And females? 12 to 15 grams per deciliter. Increased HB is called as hyperchromic anemia. Keep in mind, if it is increased, that doesn't mean you are strong. You become anemic again, even though you have more HB. Okay, that is called hyperchromic anemia. It's a condition. Okay, it's not a healthy thing. And decreased HB is called as hypochromic anemia. Very good. Methods of HB estimation: synovic hemoglobin method, oxygen chemoxygenated method, then colorimetric method, automatic method, and Sahli's method. What is the best affordable method? Sahli's method. How much time it will take to get the result out of Sahli's method? Maybe 2 minutes is for reaction. So you will maybe hardly 10 minutes. Within 10 minutes you will get the results. Is Sahli's method reliable? Will it give accurate results? Now listen. Might be our Pradeep has a color blindness. So definitely person to person the perception of comparison will be different. So this won't give accurate result, but it will give approximate result. Approximately correct result, but not accurate result. Accurate result can be given by synovic hemoglobin method and colorimetric method. Is it clear?
Yes, sir.
Yes. Now can you able to perform this test? Very simple. So first you will take a small finger prick. From finger prick you will collect blood up to 20 mark and you will transfer to the tube. So the steps in this test are take N/10 HCl up to 2 mark, then collect blood up to 40 microliters of blood, transfer to the tube, then start adding drop by drop distilled water until the color matches to the comparator color. Once it is matched, take the upper meniscus value. I hope you know what is upper meniscus, right? The upper part of the tube. Okay, the following students must go to HOD office. 23 BML 70011. Yes, then 70039. Then 70035. Three of you can go to the office. But you can you can go to the office. What are your UIAHS? 11.